In this talk, we are going to learn about different aspects of viruses. Viruses are not considered as living organisms, but instead are considered as non-living particles. Despite being non-living particles, they are known to cause sickness because they are obligate intracellular parasites. Obligate implies that when the virus is able to interact with a host cell, it will always cause a disease and not wait for the right or opportune time to infect that cell. They are intracellular because when they do cause infections, they have to enter inside a cell in order to cause that infection. That is why they are obligate intracellular parasites. Viruses are considered to be inert until they're able to come into contact with the cell that they will infect. Such a cell is called as the host cell. In order to know about the discovery of viruses, we need to know about the scientist Dmitry Ivanovsky. Dmitry Ivanovsky wanted to know what caused a disease in plants called as the tobacco mosaic disease. In this disease, the leaves of the tobacco plant showed signs of stress and they died. The tobacco mosaic disease caused a lot of problems with tobacco plants, which was a very important crop at that time. Now we are talking about the 1800s. What Ivanovsky did is he took the plant sap from these infected tobacco plants and filtered it through a special type of filter called as the Pasteur-Chamberlain filter. This filter was designed in such a way that it would trap all the bacteria so that any filtrate that comes out of this filter should not have any bacteria. Now, when Ivanovsky got the filtrate from this filter, he observed that even though it ideally should not have bacteria, that filtrate was still able to cause disease in healthy plants. So he concluded that the tobacco mosaic disease was caused by a bacteria that was very, very small and hence was able to filter through the Pasteur-Chamberlain filter, or it was caused by some toxins made by bacteria. A few years later, another scientist, Martinus Beijerinck, repeated Ivanovsky's experiment because he too wanted to find the causative agent of the tobacco mosaic disease. When he repeated the experiment, he showed the filtrate that was formed did not have any microbes even though it was contagious. Thus, he proposed that that infectious fluid had a novel pathogen in it. He was the one who coined the term virus, which means a poisonous fluid. In case you wondered what caused the tobacco mosaic disease, it is a virus called as the tobacco mosaic virus. All viruses have a capsid that is made up of identical repeating protein subunits called as capsomeres. The capsomeres provide structural symmetry to the virus. The capsid covers the genome of the virus, which is a nucleic acid that could be either DNA or RNA. The genome could also be double or single-stranded DNA or RNA. Monopartite viruses are those that have a single molecule of DNA or RNA present in the virion as part of their genome. Segmented viruses, on the other hand, have more than one molecules of DNA or RNA that are present in a single virion. In the case of multipartite virus, the genome comprises of more than one DNA or RNA molecule, but these molecules are packaged into different virions. All viruses have a capsid and genetic material. However, only some viruses have an envelope. The envelope is a phospholipid bilayer that has proteins embedded in them. The envelope is usually obtained from the host cell that the virus has infected. So let us consider these virions that have been made in a host cell. When the time comes for them to be released into the environment, they form a bud in the host cell, and the plasma membrane of the host cell now covers the capsid of that virus. And once it's released from the host cell, the mature virions now have the envelope, which is nothing but the phospholipid bilayer that they got from the host. Depending on their shape, viruses can be classified as helical viruses, icosahedral viruses, and complex viruses. Helical viruses usually have a rod or filament shaped. They can be naked or have an envelope surrounding them. And once again, this envelope is usually obtained from the host cell that the virus infected, and it is a phospholipid bilayer. An icosahedral virus has an icosahedral shape which is derived from 20 triangular faces that make the capsid. The icosahedral viruses can be simple or complex. Simple icosahedral viruses do not have an envelope, while complex icosahedral viruses tend to have an envelope surrounding the capsid. Complex viruses, on the other hand, are neither helical nor icosahedral. Another way to classify viruses is the Baltimore classification scheme, which is based on how the messenger RNA is being synthesized from the viral genome. The Baltimore classification scheme takes into account the type of nucleic acid. So viruses can either have DNA or RNA as part of their nucleic acid. It takes into account the strandedness of the nucleic acid. Are the nucleic acids single-stranded or double-stranded in the viral genome? It also takes into account whether there is sense or antisense genetic information in the viral genome. Finally, it takes into account whether the virus uses reverse transcriptase during the infection process. Viruses come in a variety of sizes and shapes. Some viruses are really small, like the West Nile virus, which has a size of 22 nanometers, while others are huge, like the Mimi virus, which is around 700 nanometers. Now, viruses can infect living organisms of every kingdom, and hence there is a whole wide variety of viruses out there. The general steps of a viral infection include interacting with the cell that the virus plans to infect. This interaction then leads to an attachment to that host cell, and once the virus has made this attachment, it can penetrate into the host cell. Now, in some cases, this penetration is not necessary, but in many instances, it does occur. The next step is uncoating of the capsid. The genetic information of the virus is inside that capsid, so for it to exit the viral capsid, there has to be an uncoating event. This uncoating event can happen either inside the cell or it can happen on the surface of the host cell. Once the uncoating has occurred and the viral genome is present in the host cell, the viral genome takes over the host machinery, and now synthesis of different viral proteins as well as viral nucleic acids occur. Now, these new viral nucleic acids can be used to synthesize additional viral proteins. Ultimately, maturation and assembly of new virions will occur, and these virions will then be released into the environment. The new virion synthesized can be released into the environment by different mechanisms. One mechanism is lysing the host cell. In this case, the viruses are able to destroy the host cell and be released from the host cell. These types of viruses are called as lytic viruses. Another mechanism is where the virions bud from the host cell. The host cell eventually will die, but it takes a little longer for the host cell to die when the virus buds from the host cell. The process of the virion budding from the host cell is a complex process and normally observed in enveloped virus. There are different steps involved, and the first step is the formation of the bud. The bud then grows, and as it starts growing and becoming larger, ultimately the membranes that are holding the bud to the host cell will fuse so that the bud can now completely separate from the host. In this case, the bud is going to be a virion. Now, some viruses have the ability of incorporating their genome into the host cell's genome. So, for example, when this virus infects the host cell, it incorporates or inserts its genome into the host cell. That viral genome finds its way to the host cell's genome and then gets incorporated so that it is now part of the host cell's genome. Now, in this situation, when the host cell replicates its genetic information, it is also replicating the viral genome in the process. During this cycle, there is no or low number of virions being formed, and this is called as the lysogenic cycle. Now, the viral genome is not present in the host genome forever. When the time is right, the viral genome will excise itself or remove itself from the host genome, and then it starts the process of making virions. Thus, virions are actively formed and packaged, and once there are enough number of virions in the host cell will eventually die because the virions will be released into the environment and it has a high viral load. This cycle is called as the lytic cycle. And thus, these types of viruses are able to exist in a lysogenic cycle as well as the lytic cycle. There are many different viruses that are present in the environment. And so, in order to explain how viruses originated in the first place, different hypotheses and models have been proposed. One such hypothesis is the virus early or the virus first hypothesis, which states that viruses were present even before living cells were formed. So they were present in the pre-cellular stage of Earth, where we had self-replicating molecules. These self-replicating molecules, which were RNA molecules, were able to be covered by proteins, and thus they were able to form the first viruses. Now, of course, with time, we were able to form cells, but at that time, viruses were already there. Another explanation is the regression hypothesis. In this hypothesis, it is considered that they were ancient free-living cells and they lost parts of their genome. And when that happened, their genome became so small that it could no longer be a free-living organism and evolved to become a virus. The only way this entity was able to replicate its genome is when it was able to infect another cell. The escaped genes hypothesis states that in normal cells, there were selfish genes that escaped, and when they escaped, they evolved to form viruses. A model that has been proposed in order to explain the origins of virus takes into account the virus early hypothesis that stated that we already have self-replicating entities around. But these self-replicating entities captured capsid genes from ancient cells. So thus, as the cells evolved, more and more genetic material was captured by these different viruses, resulting in the diversity of viruses that we see today. Thus, this is a combination of the escape genes hypothesis and the virus early hypothesis. The symbiogenic model builds on the regression hypothesis. In the symbiogenic model, the researchers proposed that some ancient cells formed symbiotic relationships with other ancient cells. Now, with time, some of these symbiotic cells lost their genetic elements. And when they did so, they started becoming more and more dependent on other cells for their survival. Eventually, they lost so much genetic information that they became parasites and hence evolved into viruses. We have viruses that can infect organisms in every kingdom of life. The first set of viruses we're going to look at are bacteriophages, which are viruses that infect bacteria. Some viruses can show both lytic and lysogenic cycles. Bacteriophages are becoming very interesting because they are being considered as therapeutics to limit bacterial growth, for example, for the treatment of infections caused by bacteria that are unable to be treated with antibiotics, or to prevent the spoilage of food so that food can last longer. Some examples of bacteriophages are the lambda virus and the T4 bacteriophage. It should be noted that there are many other types of bacteriophages that are present in the environment. Viruses that infect plants are responsible for the loss of a significant number of crops every year. There are many different plant viruses that can infect a wide variety of plants. One of the most well-studied plant viruses is the tobacco mosaic virus. This is the same virus that Martinez Beijerinck studied. The tobacco mosaic virus is known to infect not only tobacco plants, but also other types of plants like orchids and tomatoes. Examples of other types of plant viruses include the cucumber mosaic virus, the tomato yellow leaf curl virus, or the potato virus Y. Viruses that infect animals are responsible for many deaths every year. There are many different viruses present that can infect humans, and let us have a look at some of them. Smallpox is a disease that is very deadly that was caused by the smallpox virus. A vaccine was developed, and that vaccine helped in eradicating smallpox. This is the only instance where a vaccine has helped us eradicate a disease. A polio virus causes polio, and the disease polio usually results in paralysis. In fact, in the 1900s, we had so many people that were afflicted with polio, and many a times to help them through the disease, they had to be put into these chambers called as the iron lungs. A massive public campaign was initiated, and with the help of the government, a vaccine was developed. The government established an organization called as the March of Dimes, which helped in developing this vaccine. The rabies virus causes the disease rabies, which results in many neurological problems and ultimately death. The rabies vaccine that is available is unusual because it has to be taken only after an individual is bitten by the animal. Hence, it is used for treatment purposes instead of prevention purposes. The influenza virus is known to cause the flu, and the influenza virus is actually a segmented virus. We have a vaccine against the influenza virus, but because the virus mutates so regularly and changes its genome, we have to take the vaccine every year. Norovirus is a virus that is known to cause the stomach flu and is very contagious. The herpes simplex virus is known to cause cold sores. The human papilloma virus is known to cause genital warts, and in some cases, it can also cause cervical cancer. The human immunodeficiency virus, or HIV, causes a disease called as AIDS, which is acquired immunodeficiency syndrome. HIV infects a specific type of immune cell called as a helper T cell. This helper T cell plays many important roles in ensuring that our immune system functions well. Once HIV infects a helper T cell, it can remain dormant in those T cells for many years. However, when it does get activated, it can cause helper T cells in the body to die because it starts making many virions. With the loss of these helper T cells, the body's immune system becomes very weak, and usually individuals die because of other types of infections since they don't have a functional immune system. SARS-CoV-2 is a type of coronavirus which was responsible for the coronavirus disease of 2019, which is popularly known as COVID-19. It resulted in a global pandemic that we all lived through, and it brought the international community together to engineer the first messenger RNA vaccine. With this, we come to the end of our talk, where we learned about the general features of viruses, including the structure of viruses, the discovery of viruses, the way viruses can be grouped based on their shape, or how the messenger RNA is synthesized, the different steps involved in the viral infection, the origins of viruses, as well as examples of viruses that can infect organisms belonging to different kingdoms.